Hello, my name is Nawaida Binti Omar, and today I will be presenting about my research about antimicrobial characteristic of Anadara Granosa shell-based hydroxyapatite. In this presentation, I will be talking about the objective, problem statement, scopes, literature outcome, methodology, research flowchart, and some of my outcomes. The objective of this research is to measure the inhibition of bacterial growth of hydroxyapatite originating from Anadara granosa shells using various concentrations of HAP, to determine the effect of HAP on gram-positive and gram-negative bacteria, to predict if Anadara granosa shell-based hydroxyapatite is suitable to be used as biomaterial. The scope of this research is to produce hydroxyapatite powder from the shell of Anadara granosa, to synthesize HAP from HAP prefabrication technique. The HAP produced will be characterized by SRD and SAM. HAP obtained from the shell will be cultured with SRS and E. coli to observe the minimum inhibition zone. The literature outcome will cover about the introduction of hydroxyapatite, method of synthesizing hydroxyapatite, characterization of hydroxyapatite, and antimicrobial characteristic tests. HAP has the same form and composition as human heart tissue. It is hexagonal in shape and have the same stoichiometrical calcium to phosphate ratio as bone apatite, which is 1.67. HAP are the most stable calcium phosphate compound. HAP also are employed for bone repair and regeneration in many forms such as scaffold, block, granules, and also in combination with polymers or ceramic or as coating on orthopedic or dental implants. HAP can be extracted either chemically or from natural source. There are several methods of synthesizing hydroxyapatite. I will be talking about sol-gel precipitation method and wet precipitation method. The benefit of sol-gel precipitation method is the ease in fabrication, low temperature processing, fine microstructural, and chemical control. This method can generate hydroxyapatite in thin film or layer that will have large specific surface area, which is useful as carrier of drug adsorb. Sol-gel precipitation method is basically done by mixing the calcium source to the phosphate source to obtain desired calcium to phosphate ratio of 1.67. The solution is stirred slowly until the formation of gel. The gel is then dried and undergo the process of heat treatment to obtain hydroxyapatite powder. As for wet precipitation method, the benefit of wet precipitation method is the formation of safe byproduct, which is water. This method can generate HAP in form of precipitate. This method requires alkaline preparation as the source of calcium and acid as phosphate source. Then the process is followed by titration to form the precipitate, then aging and filtration, drying, and also heat treatment. The hydroxyapatite produced by this method will be in the form of spherical light. In this study, X-ray diffraction spectroscopy (XRD) and Fourier transform infrared spectroscopy (FT) will be done. XRD will measure the quantitative and qualitative analysis of crystalline phase of HAP produced, while FT will analyze the quantitative and qualitative analysis of functional group in hydroxyapatite produced. Antimicrobial characteristic test is done to spot potential medication resistance in common pathogen and to guarantee susceptibility to certain infection treatment. Broth dilution method is one of the most popular techniques that provide quantitative and qualitative results. The minimal inhibitory concentration and MIC can be determined using this technique. MIC are useful in determining the resistance level of particular bacterial strain. This is the methodology that I use in this research. Firstly, the extraction of calcium carbonate, then the production of calcium oxide from calcination, then synthesization of hydroxyapatite by wet precipitation method, and then characterization of hydroxyapatite by XRD and FT, and lastly, the antimicrobial characteristic test done, which is broth dilution method. Firstly, the seashell will wash and then dehydrate using universal oven. After that, the shell is crushed and grind to form calcium carbonate powder. After calcium carbonate powder is obtained, it will undergo calcination process to produce calcium oxide powder. Calcium oxide powder will undergo dilution to form calcium hydroxide solution that will be titrated with phosphoric acid. After the titration process ended, the solution will be left for aging to produce sediment. We will undergo centrifuge wash to form the precipitate. And the precipitate will undergo dehydration with universal oven and then muffle furnace to form hydroxyapatite cake. The hydroxyapatite cake will be grind to form hydroxyapatite powder and then will be characterized with SRD and FT to see if hydroxyapatite powder is obtained. The hydroxyapatite obtained will undergo antimicrobial test by Broth dilution method to determine the turbidity and culturing on agar. The culturing on agar will undergo incubation and then determination of minimum inhibitory concentration. The result will be then analyzed. These are some of the outcomes that I have done for this research. I already bought the shell and the shell is crushed and grind to form calcium carbonate powder. The calcium carbonate powder also already undergo calcination process in muffle furnace under 1000 degree for about 1 hour and 30 minutes to produce calcium oxide powder. So I already obtained the calcium oxide powder. I will be continuing to do the titration process after the final exam to obtain the hydroxyapatite powder that will be further characterized and undergo antimicrobial tests. That's all from me. Thank you.